Welcome to NYU Langone Insights on Psychiatry, a clinician's guide to the latest psychiatric research. I'm Dr. Thea Gallagher. Each episode, I interview a leading psychiatric researcher about how their work translates into clinical practice. Today, I'm speaking with Dr. Tanya Sippy. Dr. Sippy is an assistant professor in the Departments of Psychiatry and Neuroscience Physiology here at NYU. She's also the Principal Investigator at the Sippy Lab and Associate Director of the Psychedelic Medicine Research Training Program. In our conversation, we talk about how the brain links sensation to action, the complex role of dopamine, and the hype versus reality of psychedelics.
Well, thank you so much for being with us today, Dr. Sippy.
Thanks for having me.
So we're just going to talk a little bit broad, and then we'll get more specific as the episode goes on. But can you tell us a little bit about your current research?
Sure. We actually have 3 research projects in my lab. 2 of which are funded by the NIH, and the third's a bit of a wild card, but I'm sure will be funded by the NIH soon. So in the first project, we study the process in the brain that actually links sensation to action. And so when I say sensation, I mean things in the environment. So an example that my postdoc gave yesterday, actually in our group meeting talk, was that when she moved to New York, she realized that she needed to actually navigate the streets in a very different way than she had before. So it wasn't good enough just to look at the crosswalk sign. She also had to pay attention to the honking taxis and the people coming at her from all directions to safely navigate across the street. And that's the central process that we're interested in because I really agree with Daniel Wolpert, who actually is a computational neuroscientist who I got to see speak last week, and he really thinks the brain is to create movement. I think it's probably broader than that, but I think movement is an essential component. So I also think that linking that movement to things in our environment is a fundamental process. It's very important, and it underlies a lot of neuropsychiatric disease. And so to study that, we look at a region of the brain called the basal ganglia. It's very well conserved from rodents all the way to humans, and we study it in rodents. And the input nucleus of the basal ganglia is this area called the striatum. It's actually gigantic in mice, very, very big in humans as well, but bigger in mice, proportionally speaking. And it actually gets a lot of inputs from sensory areas all throughout the brain, and those inputs actually aren't very well studied. And so we know that the basal ganglia is important for motor output because lesions in the basal ganglia, so Huntington's disease and Parkinson's disease, people are movement disorders. But we don't understand how sensory input influences that process. So that's one project in the lab. And I have 2 more I could talk about.
And can you just tell us a little bit about how you study these things?
Yeah, so my lab uses a variety of really cutting-edge approaches, I think. I spent a long time learning these approaches across different laboratories across the world, including Los Angeles, New York, and Switzerland. And basically, the main technique that we use is called electrophysiology. Electrophysiology is used in many labs around the world. It's used in humans, non-human primates, rodents, basically any organism you can think of, any model organism. But we take a rather unique approach to this in that rather than recording from the outside of cells, and kind of looking at their firing patterns, which you can do in a way that's extracellular. We actually record from these neurons intracellularly while the mouse is awake and doing behavior. And in that way, we can actually look at not only the firing of the neurons, which is kind of the tip of the iceberg, but we can look at all of the activity, which we call subthreshold activity that leads to that firing. And that lets us look at really important cellular properties that no one else can access, really.
And what is the big question that you're hoping maybe to answer, or the small question that you're hoping to answer with this project particularly?
So we really want to understand how learning changes sensory representation. And we want to understand how that happens in a normal model. And then we want to understand how that process goes awry. For example, let's say in addiction, where often what addiction is, well, it's very complicated, but one thing that really leads to relapse is that you have an overrepresentation of things in the environment. So let's say, you walk into a bar and you see people there and you see the glasses and you hear the cheers and you smell the alcohol. Those are all things that might lead somebody, even if they don't want to drink. Same thing you can imagine for smoking or really any drug. And so we'd like to understand first how this happens in a normal setting and then understand how it goes awry in these different conditions.
And can you tell us a little bit about the 2 other projects?
Yeah. So the second project, also NIH funded, is one basically where we're really trying to find out what is the function of dopamine. And I know that sounds like an age-old question because it is indeed an age-old question. We've known about dopamine for 100 years, so the better part of a century, maybe not quite 100 years. And it was really Arvid Carlsson in 1957 who really described that dopamine is not a precursor to other neuromodulators. It itself is a neuromodulator. And that's when we really realized that, dopamine is doing something aside from being a precursor. And it really was also Peter Milner with James Olds, and this was in 1953. They really started to do these self-stimulation experiments in rodents where they realized that if the rodents were able to self-stimulate their brains, where the dopaminergic cell bodies were, that they would do anything for this self-stimulation. So press a lever many, many times. And so this was a pleasurable phenomenon. So then, I think we knew as a field, I wasn't alive then, but I think the field knew at that time that dopamine might have something to do with reward. In 1996, Wolfram Schultz and his colleagues, they actually showed in non-human primates that dopamine neurons, when you record from them, do something very interesting. And that is that if you pair dopamine with an unexpected reward, or if you pair a sensory cue with an unexpected reward, what you'll see is an increase in dopaminergic firing. We call that a phasic firing because it happens briefly. So you see a brief increase in the firing of the neurons and then it goes back down. And you'll see that at the time of the reward if the reward is unexpected. So let's say, the animal or the human hasn't learned that the sensory cue predicts the reward. You'll see the dopamine at the time of the reward. If you then pair with the sensory cue over enough trials and the animal learns that that they're going to get a reward after the sensory cue, dopamine neurons do something very interesting in that they fire at the time of the sensory cue and they no longer fire at the time of the reward. So when they know the reward is coming, dopamine neurons fire, right? And then the last thing that they showed in this landmark paper is that if you then omit the reward, so you have the sensory stimulus and, you're expecting to get a reward, but it's omitted, the dopamine neurons will actually pause their firing. So you see a dip in their firing. So this was very interesting, and it led to this idea called reward prediction error, and also like a huge concept in the field called reinforcement learning. And this is based on the idea that what dopamine is doing is signaling this reward prediction error. So it's not so much that it signals reward, it signals the difference between an actual reward and expected reward. And that's basically how we learn from rewards in the environment. So that story goes on. So dopamine is not actually, we don't know what it's doing yet. So we think, it's for reward prediction error. That was, again, I told you, and this is 1996. So you might say, okay, the book is closed. Why are you still studying dopamine? Well, the thing is, just because, dopamine, correlates with these RPEs and you can do many other, causal experiments, it doesn't mean that we know how this process is actually working in the brain. So we don't actually know what dopamine is doing to the downstream neurons, which happen to be in the striatum, which is the nucleus I told you about before that we study in my lab. We don't actually know what it does to those neurons to allow for humans or animals, for that matter, to learn. And so that's what this second project is about.
And so if we did know more about that, how would that maybe move like the research forward?
So it would move the research forward, I think, in a number of ways. So I think if we could understand what are the downstream effects of dopamine, we could start to understand, for example, in addiction, what are the neurons that are actually responsible that are leading to the use, and how can we then modulate their activity, not by just targeting dopamine, but more specifically targeting the neurons that are responsible for mediating the behaviors that dopamine is enabling. So that's one way, but many, many other ways as well.
Yeah, it sounds like you're saying dopamine isn't in a vacuum. There are a lot of other things it's connecting to that you're trying to understand because maybe you can impact parts of the cycle or parts of the pattern.
Exactly. Dopamine, again, and it might even be this red herring. It's so many studies are focused on dopamine, and you get to the end of the study and it's like, oh, yes, dopamine correlates with this, or yes, dopamine does this, but it has to be. Our brain is not just dopamine. If it was, I wouldn't be speaking to you right now. It has some elements, but there's so much more. And I think that's what we really need to understand.
I think you said there was a third project you wanted to discuss?
Yeah, so this project directly stems from my work as a psychiatrist. So when I was an intern here at NYU, it was a very difficult transition to make for many reasons. I went from being in a Swiss lab to being in Bellevue Hospital, and those are quite different environments. And one of the things that I was doing quite a bit was dealing with very, very, very sick patients, and actually. Something that I was really reckoning with quite a bit was how difficult it is to give patients the news that their life is going to end. And that was something that took a huge emotional toll on me, and really made me question whether or not, I wanted to actually finish residency because it was so, emotionally burdensome. And I wasn't sure if I was the person who should be in that role. So I came back from work, one day, and my husband knew that this was a very difficult time for me. And, he asked me if I had read this article in the New Yorker. And I said, no, of course not. I don't have time really right now to be reading the New Yorker. And he told me about a doctor named Stephen Ross, who had spent a great deal of time actually getting a clinical trial off the ground at NYU, where he was looking to treat patients with terminal diagnoses with psilocybin to help them with their anxiety. And I thought that this was just a beautiful demonstration of how pharmacology could make a huge difference in someone's life at a time where they really, really need it, and in a way that is super meaningful. So luckily for me, Steve or Dr. Ross was working on my floor when I was on 20 West doing a psychiatry rotation. And, he's very open. And I knocked on his door and just started chatting with him about this work and how I could possibly be involved. And we started a psychedelic research interest group for residents. And then fast forward about 4 years when I started my lab, the Center for Psychedelic Medicine was established at NYU. And they were looking for someone to kind of help them with a training program that they had set up with the generous support of MindMed. And they asked me to do it. And one of the postdocs in the program, who was actually a research assistant professor, decided to come to my lab and start doing preclinical work to understand what the psychedelics are doing in the brain. So she came. Her name is Sarah Menenga. We got the appropriate licenses. That took 2 years. Oh, yeah. And now we have research underway that's really investigating whether or not rodents are an appropriate model to understand how the psychedelics are working in the brain. And so that's kind of where we are with that project. And I think it has great potential. And I also have a graduate student named Jael Guardado who's working on this now.
So again, you're kind of looking at more of the granular level, trying to understand the mechanisms and trying to understand how it works?
Absolutely. We do not know what this particular serotonin receptor that the psychedelics work on is doing. It's called the serotonin 5-HT2A receptors, many other receptors as well. And we don't know if it's that receptor, other receptors, or how these drugs have their therapeutic effects. And we really don't understand, even in the acute scenario with what these drugs are doing acutely, how that relates to what their therapeutic effects are later. And that's something that we can very much track in a rodent, because the tools for tracking rodents nowadays have exploded. I mean, you have these deep learning algorithms now that can really track rodent behavior at a level of detail we couldn't do before. So the time is really right to understand what are these drugs doing, and then relate that to what their long-term effects might be.
Going back to that time you were talking about when you were giving people this news or talking with them, I'm just curious, what was the hardest part? Was it like seeing other people struggle so much with the fear around the concept of their own death?
When you tell someone that they are going to die, you realize that no matter who they are, they don't actually want to die. And I think that realization was the hardest for me.
Yeah, and seeing like the realization for yourself or watching them experience that, or combination?
Both, definitely. It's like, what do you say in that scenario? And you realize like there is there's no magic sentence, and there's no hug or look or smile or anything you can do in that moment. But I strongly feel that if you can give them an experience, like you can potentially with a psychedelic, that gets them out of the context of themselves, which is really what these drugs do, and rethink about the news that they've been told in a totally different way, in a totally different brain state, you have the potential for them to come potentially, to an understanding and maybe an acceptance of what's going to happen.
Yeah. I mean, I think all the psilocybin research is so fascinating. And, I work with patients with anxiety, severe anxiety, PTSD. And I think this concept, or it sounds like what I'm interpreting from what a lot of people are saying, is that for people to get like a little bit of space away, to get a little bit of perspective, what we try to teach people to do in mindfulness, but can be so difficult, when you have anxiety and stress. But for people to get that space where they can see their thoughts almost like a movie, where they can really kind of disconnect from them or not feel so, overwhelmed by them. I feel like that's such a fascinating part of what's happening in the research.
Absolutely. And, I'm not sure that rodents are the best model for all of that, but I do think that, they are models for many things that we can study that are related to that process. Yeah.
Mm-hmm. Well, it sounds like what a lot of your research is saying is that we understand these things work, but it's really important to get a better sense of how they're working and the intricacies that exist.
Yeah. And also, establish the safety profile, the dosing, how often, in what setting. I think, with animals, we can do this research relatively quickly. And if the drugs are doing similar things to the animals, then we should be able to study all of these things quite well and use what we've learned to apply them.
Yeah, it's interesting from my interview with Dr. Bogenschutz. He said that a lot of patients had this similar experience. And just kind of with what you're talking about, he said that a lot of them reported this experience was intense, memorable, and meaningful. And I think that's it's going to be so interesting to see how that all continues to play out. And it's amazing that you're doing the research, too, to kind of understand more of what it's exactly doing as well.
Yeah.
So personally, why is this such an exciting or interesting area for you?
Which one?
I guess let's go with the dopamine.
So dopamine is... So I have also like a long, complicated relationship with dopamine, I would say. I think that ultimately, potentially, dopamine is a large reason. I'm not sure. Maybe I was born a scientist. I have no idea. Neither of my parents are Nobel Prize winners. These like scientists where like, their parents were Nobel Prize winners. And then you're like, this was just epigenetic, like in your destiny. You see them like halfway through their career and you're like, yeah, you're also definitely getting the Nobel. I'm not in that boat, I would say. So, but at some point in college, so when I was in university, I knew, I wanted to be a doctor. I wasn't sure like what element the science was going to play. I ended up getting started in a lab where I was just doing synaptic transmission. And it's fascinating, actually, area of research, just how two neurons communicate with one another. Ultimately, when I was in graduate school, that wasn't actually sufficient anymore. Like things had become a little bit more, you grow older, and I realized I was doing an MD PhD. And at some point, I was looking for, okay, like what is really meaningful for me, to study. And dopamine really came up as, something that could be interesting. And the reason why is because this was just years after Wolfram Schultz's work. And I was like, okay, dopamine is like reward and pleasure in the brain. And that's what I want to study. I want to study reward and pleasure. So that was like 2007 and 8. And even like my Twitter handle at the time said like, I'm a scientist who prefers not to limit experiences which allow for the release of dopamine in my head. That's where I was, 2008, 9. That stayed there actually for many years. I just removed it recently. And the reason I removed it is because it is too simplistic of a view of dopamine. So I knew that I wanted to study it. But then, as I started actually doing work in the striatum, and this was my work in Switzerland in the lab of Carl Peterson, where I was actually studying the function of striatal neurons. During that time, and then also as I transitioned to faculty here at NYU, I realized that dopamine is so much more than a reward neuromodulator. So dopamine is actually active in a lot of different scenarios when we encounter anything that's novel. certainly for motivation and learning. I think that's been well established, and we've already talked about that quite a bit. But the other thing that dopamine is really, really important for is movement. And that's how these projects kind of tie together. So in Parkinson's disease, when you have a loss of dopaminergic function, patients cannot move. And so there's been a lot of work actually by, some people at NYU, including Nicholas Trich, but also Rui Costa and Josh Dudman and many others out there, Anatol Kravitz. I mean, I can go on and on. Anatol Kreitzer, that is, and Lex Kravitz. But many, many people who have studied the role of dopamine actually in other functions and other behaviors, and one of those is just movement. And so what this has really brought up is that, okay, dopamine is active during reward, right? It is active during movement. And how does this neuromodulator accomplish these two functions when all the cell bodies are in the same place and projecting to, different areas, but there's a lot of overlap, right? So how is it that in one context, dopamine can be doing movement, and in another context, dopamine is doing reward? And this is where I think the field is at. There's actually a scientist named Vanessa Ruda at Rockefeller who does work in Drosophila, and one of her most recent papers is really just looking at this, like how can dopamine neurons actually accomplish these two jobs? So I think that's kind of like a very exciting direction and something that I'm really interested in, and many neuroscientists are. And I think for now, that's kind of why I study it, because it's both involved in the first project and movement, but also by itself, I do think, studying reward in the brain is also still exciting. So now it captures, all of this basically.
And when you're talking about, movement and pleasure, do you see them as two separate functions, or are there any crossover?
I think there's a lot of crossover. I mean, I think in order to move, you need to be motivated. At some level. And so I think, a lot of people would say that. So I think there probably is crossover. But when you actually look at the time scale of dopamine signaling, and this is something, a lot of labs are starting to do. There's a new tool actually that was disseminated by a couple of labs. Actually, we use the one by Luke and Lowe, and basically this is a sensor actually that you can actually put into the brain. And you can now, at least in rodents, and certainly you'll be able to do this in other organisms as well, you can actually track the release of dopamine in real time. And this is revolutionizing the field basically, because now you can actually see real time during behavior fluctuations in dopamine. And what we're realizing, and this is actually more work actually by Nicholas Trich, who's my colleague here at NYU, and his graduate student Anya Kroc, is that dopamine is actually fluctuating all the time. So not just during movement, but also in periods where, the animal is just stationary. You see dopamine going, up and down. And you also see, as many people have reported for many years, this phasic increase in dopamine during reward. So we're understanding that dopamine release is much more complex, and it might be accomplishing different things in just the timing or the way that it's released onto different subpopulations.
And that would be pretty fascinating also with people with various mental health conditions, right? Like ADHD or bipolar, understanding, what is maybe the impetus for the dopamine or the relationship to the mental health condition. Yeah,
I mean, dopamine is probably involved in almost every psychiatric disorder you can think of. To some extent. It's probably related. Again, I'm not sure if it's, red herring or if it's actually involved. It's really, really hard to know in psychiatric conditions. And maybe we're giving dopamine a bit too much credit. I mean, obviously, dopamine is a major target of many of the drugs that we use in psychiatry. Interestingly, mostly for psychotic disorders. And there is where you really see the crossover, okay? So you have a patient who is psychotic. Maybe they're manic, right? So maybe, they have like too much energy. If anything, maybe their reward systems are in overdrive. We give them an antipsychotic, and what happens? Not only do those systems come down to baseline, which is what we want so they can be functional, but they also have terrible movement disorders. And there you see the crossover of how dopamine is really important for both of these things. But it's also used, some of the antidepressants target the dopaminergic system, much less so. Of course, serotonin And how serotonin and dopamine are working together, we have this is a field that we are embarrassingly far behind in, I would say. How neuromodulators are working together, and especially these two, dopamine and serotonin. This is a project we're likely going to take on in my lab. But I think, many other labs are going to start to become interested in just how neuromodulators are working together. Norepinephrine, for example. So just really understanding this problem of they're released at these times, but what are they doing to downstream neurons, and how can we use that to better target therapies?
And do we understand anything about the serotonin-dopamine connection?
So we're starting to. So again, there was a paper actually published last month in Nature in 2023 from a young scientist, actually up and coming. Her name is Zeynep Okray, and she comes from Scott Waddell's lab. And basically, they were looking at the interaction of the dopamine and serotonergic system while basically looking at a sensory learning task, basically also on Drosophila. There's been other work as well, for sure, but this is the most recent example I can think of, and a really beautiful demonstration of how the two systems might be interacting.
Mm-hmm. But like you said, there's more to learn, and it'll probably inform, like you said, clinical care in the future.
Absolutely. Yeah. And these tools, we really can use them very, very well in animal models. So if you really want to track in a single brain area in real time release of dopamine and serotonin, at this point, that work, it's really best to do it in animal models.
When you're talking about the psilocybin, I think it's so interesting how your story of how you actually kind of came into being interested in that research. And it seems like it started with a very human experience, connecting with other humans. And then it sounds like you find a lot of value in doing the work at the, I don't know if we say like the rodent level, but at the micro level. Can you tell us about how those feel connected to you?
Yeah, I think that's kind of, going to be a lifetime of work in, my own sort of personal journey, and then also, in my experiences. It's slow. Would like to speed it up. I'm not getting any younger, I would say. But right now, I see, I do see these times where I have these big leaps of understanding, how these things are connected. The perfect example of that is when Michael Bogenschutz and Stephen Ross, we discussed that, I would be a part of the center. And then we started doing this work in my lab. That happened in the first year I started my lab, right? This was something I was thinking to myself, I'll do this 10 years from now. I'll do this 20. Once I have tenure, I'm not worried about where money is coming from. I'll just do this work. And, it happened in year one. So there was one major leap there. I do have plans, for sure, to link these things together, I think even more concretely. But we'll see how those unfold in the next few years. I think for now, I'd really like to get a handle scientifically on what these compounds are doing in the brain, and then take it from there.
And people are so excited about psilocybin, right? Like everyone's talking about it. Everyone's really excited. And you talked about how it took even 2 years to get the license, and we're learning more about the dosing. What is your kind of advice or thoughts about, I think clinicians are excited to be able to hopefully, recommend this in the future. But I think we have to obviously be patient with this process. So yeah, what are your thoughts about kind of the pace that it's moving, the excitement? Do people have to dial it back, or, we need more time? What are you thinking?
I'm so happy you asked this question. I think people need to dial it back for sure. Like, I think, you do need, look at, for example, diversity, equity, inclusion. Okay, it's a perfect example. And it's just, these things are so important. They're so fundamentally important, right? And I want to be very, very careful with how I approach this. I absolutely am a supporter of diversity, equity, inclusion. I sit on many committees. I think it is fantastic. And you need the world to get behind, certain concepts and certain ideas that are important for our society as a whole. But you also don't want to get to a point where you're stifling people's opinions, people who might, have a little bit of, but what about this side of the argument, and that's, I think it's important to at least hear those people out. We are in a country where, you should be able to have freedom of thought and freedom of speech, not so much that it harms other people, right? But I just worry that when the pendulum swings so far in one direction that maybe we are silencing people who might have, very interesting opinions on the subject, but don't necessarily agree with the vast majority. And again, like I said, I'm very much a champion, but I worry that my own strong thoughts on the subject might be alienating some people. And I just started realizing this recently, actually. So I think the same thing goes, with psychedelics or, any major thing that, has the power to change the world. There's always the other side of it, right? And I think for psychedelics, I very, very, very much worry about the way the drugs are being commercialized and the way they're going to be given to people. And I think for, the psychedelics and the dissociative anesthetics, for that matter, I really hope that the people who are prescribing these drugs are the ones who actually know what they're doing and are doing it for the right reasons, and the right reasons are to change people's lives and, to do it in a way that, is important for the person. And that it's a good match between what the person's needs are and what the drug is doing. And that it should be done with that intention. And so I think that's my, and again, I'm not trying to, put that opinion on anyone else who might have, strong ideas of how this should be done. But I'm just going to say that even for me, who's, a trained MD, a trained PhD, I really approach this with caution. And I myself would want to do it from a place where, I'm doing the research and I understand the safety before I were ever advise or, try and give this to a patient of mine.
Yeah. And I think you're saying like we have all this exciting information, but we have to do good work and good science and really make sure that we know exactly what we're doing. And if we rush too soon, we might, it sounds like it could get haphazard and then we don't actually understand what exactly is working, how it's working. And that's what you're studying, really.
Look at what is happening with THC, right? Or, CBD is fine. More like, THC, I'd say. Like, again, I'm not, judging, I think it's, I think it is what it is, right? And, for people to be able to, have an access to a drug that could be therapeutic is a good thing, right? But where are the regulations? Like who, what age should you be able to, buy, this substance, and like under what conditions should you be able to use it? And like, I'm worried that the legislation has not caught up to the legality. And this would be, I think, even worse for, this class of drugs. So I think I would like to see all the, litigation in place, or at least have, there should be a licensing credentialed process that, clinicians have to go through before they can prescribe, right? And it cannot just be any MD who has a Schedule I license. Like, this is not going to go well, right? That's sort of, I'm going to get off my soapbox now, but that's my opinion on the subject.
Well, I think it's important. I think what this research is fairly new, right? In some ways, like looking at psilocybin, ketamine, THC. And again, everyone's really excited about the work that's coming out. It all seems very positive, but I think pumping the brakes for caution and understanding a little bit more about how these work in the brain and at what dosage, what level, what age, long-term effects. It seems like that's also super important, because in history there, you can get excited about something and then find out that there are like other kind of problems that maybe weren't seen at the beginning.
Yeah, and also just a thorough screen of the patient, right? I mean, someone who really has seen hundreds of bipolar, or whatever it is, mood disorders, and can identify that, looking at the history, talking to their family, making sure that this is a person who we're going to actually do right by, and that these are actually going to be therapeutic and not harmful. That is so important.
In talking about that, with clinicians, what do you think is the best road for clinicians to become apprised of what's happening in the research, or to become more competent? Are there trainings? Are there, and maybe, this is a question, I don't know if you can answer, but are there ways for clinicians to kind of keep up and know really, like, not just kind of like the clickbait articles, but really know what's actually happening and how they could maybe get to a place where they could be a Michael Bogenschutz, helping people?
I think psychiatric training is very important. I think at least, for a psychiatrist, again, or at least that they would be involved with the the whole process, because a psychiatrist, again, can do the appropriate screening, ask the appropriate questions. And some of this is pattern matching, right? A history of psychosis. Like somebody might come to you and they're not psychotic, right? But the only way you would know is to do the appropriate questions and background search. And I think, psychiatrists are very much in tune to that type of thing, and they're very comfortable with asking those questions and finding that history out. So I think that is extremely important. And then I think, for let's say, potentially at some point, this could be a family practice doctor or, an anesthesiologist, let's say. It makes me a little bit less comfortable, but I think it could be, right? But there, I think you would need, extensive training and, what are these drugs doing? What to expect? What are the effects on the body? Not just the mind. And also, I think probably specific trainings for each substance. MDMA is not going to be the same as psilocybin, which is not going to be the same as the next thing. So I think, kind of thorough and very well thought out trainings would also be something that I would be really supportive of.
Mm-hmm. Yeah. And I think, and also like an understanding of any potential interaction effects and such.
Absolutely.
Mm-hmm.
Yeah.
Yeah. And to kind of go back to people being faced with their own death, do you think that, I'm just thinking about someone who is in that place, right? And obviously could be really terrifying. Is there a place for ultimately like some sort of compassionate use like in the future? So these people aren't waiting 50 more years. So for something like that to be allowable.
I would hope so. This is a better question for Michael Bogenschutz and Stephen Ross. I know that it could be possible in like a limited way for clinicians to apply for licenses to use these drugs as an extension of current clinical trials. I myself have not done this, but I know that, I had spoken to, I think one of them previously about this being, a possibility, but that would not reach a very large population. So the only way I think to do that is, continue doing the research, continue, having open conversations, with the FDA and with the government. Just everyone getting on the same page about what would the benefits be for these types of drugs. And I think that's the proper way forward. And it will take time, but I do think we will have these moments where, we make a lot of progress and then hopefully, get there. My hope is within the next 10 years.
Yeah. And that leads me to my next kind of question. What are you hoping to see from your research or, in the next 5 to 10 years?
I have different goals for each project, but I think I would like to have a very good understanding, at least of the basal ganglia and associated circuits that are important for linking sensation to action in the normal physiological setting. And that's going to involve, all the approaches, that I've told you about earlier. And it's also going to be, a combination of these two projects because you can't just look at, the striatum alone. You have to look at inputs coming from other brain areas. And you also have to look at the role of dopamine. So that would be really wonderful. And ideally, if we could then apply that to a disease model, such as, addiction, which is you can study very, very well, actually, in rodents. That would be, really great. And on the side of the psychedelics, I would like to use everything that my lab has learned from those two projects to really establish powerful biomarkers for what the psychedelics might be doing, and then study those processes during and before psychedelic treatment.
It seems like more and more patients are kind of they have access to the internet. They're very interested in how things are working. I feel like more and more patients want to be educated, want to understand. They don't just want to be told by a doctor, here, take this, do this. And so I'm thinking in connecting the work that you're doing, because I've even learned so much just talking to you, for clinicians who are, working with patients every day, and patients are wanting to understand more as well. Where would you advise, clinicians to go to kind of continue to learn and to learn maybe more about these mechanisms and how some of these, newer medications are working, or, where would you advise them to go to kind of continue to grow in that area?
That's a very difficult question. It might be the hardest question you've asked me, which is, sad in a way, I have to say. But I personally trust very few people with being able to advise the public on these drugs. So what we really need is we need more people who are very, very capable in this area, who are, psychiatrists or doctors doing research, scientists. We just need more of those doing this work, who are willing to do the work, but also talk to people about the work. And the only way to do that is in mass number, because each of us has, only a certain amount of time to dedicate to public outreach or to dedicate to, seeing our own patients. And so if we can get, I don't know, at least, 10,000 or 20,000 of us across, maybe those numbers are too big, I'm not sure, but basically, a critical mass, then I think, people will have many people to go to. You'll have, 12 or 20 in New York, who you can talk to. And then, we collaborate on, establishing, a place where we can put information that we really think is important and relevant. And, of course, you have very, very, very somewhat, you can edit this out maybe, but you have some, reputable news outlets, of course, as well, who are trying to responsibly publish articles on this that I think the public is looking into. But I think those articles tend to be, have either one side or the other. And depending on what, the state of mind the person is in when they're reading the article, it's either going to feed into their fear or feed into their excitement, but not actually get them, the real information in an unbiased way. So that's my worry about those articles. Although I think they're fantastic to kind of spread the knowledge that this work is happening. I think it has its limitations.
Yeah, but it sounds like reading the publications of the people that you have referred to in this podcast and kind of getting back to the studies that have happened and that we know about, and taking all of that information, it sounds like also with a grain of salt, not the research study itself, but, what the plans are for the future, because we don't have tons of studies yet.
Yes, exactly. So I think, we definitely need more studies, but let's just look at ketamine as an example, right? So, ketamine, I think, is a very, very interesting drug, right? It's one of the only drugs that we have right now in psychiatry that can actually treat acutely depression and potentially suicidal ideation in an acute setting. So that is powerful, right? Mm-hmm. Now, there are a lot of ketamine clinics in the city, right? Many capable doctors who have used the drug, who are now using it for mental health, for example, anesthesiologists, and mostly anesthesiologists, actually. But I just think that with the emerging research, we will realize that there is so much more that you can do with this drug if you actually go into it thinking you're going to treat a mental health problem. Potentially provide the right environment, provide the right therapist, all in combination. I just think that that's the direction that this is going. And I'm happy that, people can go and get the treatment, because that by itself probably is helping. But it's just an example of how much further you could potentially go with these treatments, I think, with the right team.
Mm-hmm. Yeah, I didn't know that ketamine clinics were run by anesthesiologists. I just assumed psychiatrists.
Some are run by psychiatrists, and in fact, a friend and colleague of mine is doing ketamine-assisted therapy, and I'm confident she's doing a great job. But many are not. Many are run by anesthesiologists, and many are run by people who don't have medical degrees at all, but they hire a team. And I don't know. I'm not saying that they're not qualified or they're not great centers. I'm just saying that I just don't know. And I know that there are many of them. And there's very few psychiatrists who I know that are doing, this treatment in their offices.
Yeah. And so one of the themes I think that we've talked about is this is all exciting. There's a lot to keep looking forward to, but proceed with caution a bit.
Definitely.
Well, thank you so much. This was a really exciting conversation, and looking forward to hearing and seeing all that you continue to do.
All right. Thank you so much.
Thanks so much again for that conversation, Dr. Sippy. If you enjoyed this episode, be sure to rate and subscribe to NYU Langone Insights on Psychiatry on your podcast app. For the Department of Psychiatry at NYU Langone, I'm Dr. Thea Gallagher. See you next time.